speaker is Sumitabha Brahmachari from Rice University.
Okay, while it shows up, thank you for giving me the opportunity to talk. The original idea was to only talk about bacteria, but seeing all this very cool work in eukaryotes, I couldn't resist the temptation to tell you a little bit about the things that we have been also working on. So with that, basically, we have learned again and again that chromosome has a structure. Chromosome also has a chemical state associated with it, and it's the structure and the chemical state is highly correlated to the extent that you can actually, it's predictive. You can take the epigenetic state and work and actually predict features of the three-dimensional structure. So basically, this physicochemical state, so to speak, is what is strongly correlated with structure to the extent of, and the question is whether it is driving structure or not is always an open question, and in some cases you find one aspect versus the other, and that's really one of the crux of the question, is to understand how that physicochemical state is driving structure, but it's not really a monolith. If you're thinking about this, is a very highly dynamic, it goes through, is continuously changing, and this change, what the cell wants to do is to make it very controlled. You cannot move from solid-like to liquid-like randomly. It has to be very controlled and highly active process. And the fact that it is more plastic is necessary because if it's related to function in chromosomes, function is not unique as opposed to proteins, which usually you think about one protein, one function. Chromosome has a wide range of functions. So this plasticity is actually very important to accommodate various kinds of functions, which might be actually, in different stages of a chromosome can be very opposite to each other. So the kind of simplified framework that I like to think about is like during the dynamics of this state, it's going through some functional changes, and as it's going through these state changes during this route, what are the factors and what are the underlying mechanisms that are regulating these processes? That's really the goal of modeling, to understand these kinds of aspects. And the kind of experiments which are important for this physicochemical state is the Hi-C that we use a lot. I'm not going to tell you about Hi-C, but somebody just walked into the room. It's basically a pairwise contact map. But okay, so we can move on from here. But learning from the features of the Hi-C, like off-diagonal features like compartments and loops and domains, what we have, and of course, being inspired by a whole lot of work from all different groups, just named a few here. So we came up with this kind of an energy landscape formalism. Starting with the basic idea that it's a polymer, which to some extent is fine, but it's not constrained enough. Just having a polymer doesn't give you all the features you see in a chromosome. You need to add additional biases to this structure, and these biases can come in various flavors, is what basically we worked on, is to see like what are the typical kinds of biases that exist, and then sort of grouping them together to understand the physics of the problem. And one of the main differentiating things here is to realize what potentials are acting intra-chain, like this, what we call lengthwise compaction, is basically an effective way of calculating recapitulating loop extrusion. So these are controlled by these proteins, and the other kind of phase separation, which doesn't really care about the chain. It's just based on proximity. So that competition between intra-chain and something that doesn't care about inter and intra actually leads to a very wide diversity of phenotypes in structures that we actually see. So using this framework, we have been able to explain some very interesting features, such as what gives rise to chromosome territories, what terms are giving rise to features like chromosome territories, and what terms are giving rise to features like centromere clusters. So one of the phenotypes that we saw in a talk yesterday was this east centromeric phenotypes, centromeric clusters, and what we saw is like there is a competition between these terms that can drive one phenotype versus the other, and these are specific proteins. If you deplete these proteins, you can see a very quantitative shift of this phenotype from one to another, so this is predictive in that sense. And by looking at various organisms across the tree of life for eukaryotes, we basically saw that all the large-scale average features that we see can be recapitulated within this kind of a framework where you have certain generalized forces which are driving the structure of the chromosome. So this was fine, and we went one more step. So beyond this, where we included, beyond just equilibrium, we included the activity, it included non-equilibrium motorized forces. The idea is slightly different, but Madan presented about active stresses. Here, the idea is that the active stress has a memory. So it remembers because the motors bind and pull in some directions, in certain direction over some time before unbinds. So there is a memory component to the active stress. Which leads to variety of features. We explain, we go into this. Some of the very non-intuitive things we find is things like phase separation, which we usually think in terms of Flory-Huggins kind of sticking, is not strictly necessary in an active paradigm. You can get into kinetically trapped states where purely repulsive things can actually form something that looks like a condensed state. So those things can happen without an effective attractive interactions. These are very novel kind of phases where entanglement between the polymer is important in that scenario. And similarly, like features like fountains can happen. So this is like, if you add activity to certain regions, so this is a phase separated polymer. If you add activity, these are much more dynamic. So they sort of come out and you form these hairpin-like structures which lead to these features of sort of hairpin-like. And the most interesting thing is if you look at the MSD exponent across the cell, you see this small bump. Now you might want to ignore it, it's too small, you really care about this, but if you don't have motorized forces, you will never be able to recapitulate this bump because that's the super diffusive signature. So okay, so we have been doing all this, and this framework has been interesting to recapitulate many features of eukaryotes. But if we move to more simpler organisms like bacteria, much smaller chromosome, it turns out because they don't have the compartmental patterns, one of the main feature is absent. So we need to rethink a little bit about this underlying framework where, think about chromosomes that do not have compartments. So in this case, we can still use the same energy landscape, but the bias needs to be rethought. And so the one way to do this is to find a generic bias, which is irrespective of any underlying mechanistic bias. Because there is no, if we don't know what the mechanism is that driving a particular structure, we can still try to recapitulate the structure. The crux of the problem is, we can have a polymer, we can, under some potential, we can calculate what the probability of contact is. Basically the constraint is the simulated contact should be equal to the experimental contact, and whatever is that potential should be a good representation of the bias. The way to find this, I'm not going into the details of this, but you can invert the problem and say, like, let me find the probability distribution by using the principle of maximum entropy. And I'm not going to talk too much about this, but what's very interesting, this paper by E.T. Janes, he basically proposed this. He says that if you use this model, basically I'm paraphrasing what is written, if you use this model and you find out that your model is inconsistent with experiments, it doesn't really mean that your model is wrong. It basically means you have not incorporated all the constraints that are present in the system. So you really need to think about what are the extra constraints that are present in the system. Okay, so by using this, we can come up with a bias potential and do an iterative algorithm to find out what the underlying bias potential is. I am again glossing over all that details. If you're interested, I also have a poster. I'll be happy to tell you about this, and it's also on the archive. The work is on the archive now. So this is showing you that iterative process. What I'm showing you here is the experiment. This is simulation. I start with a homopolymer, so there is no feature. This is the optimized force field. So this is the bias potential. This is the scaling experiment simulation. And this is over number of iterations, how the mean error is going down during iterations. And I'll show this as a GIF, and it's going through it's basically going through iterations, and it's learning what the underlying bias is from the data. And then, after some number of iterations, you can stop this and say, okay, this is my minimally restrained bias potential. And using that, one can do the simulations to find, this is simulated high CMAP and get ensembles of structures. So that's fine. We see that these ensembles of structures are very consistent with high CMAP. We don't have any underlying mechanistic bias. These are just structures that are directly derived. But what do we do with it, right? And what are the okay? So what's the question, right? The question is, we want to understand co-replication segregation of bacteria. Bacteria has replication and segregation happening at the same time. We'll use Bacillus subtilis as an example. So the question is, bacteria during exponential growth, bacteria really needs to pull the two ori into two opposite poles. You need some kind of a polar force. It's much faster than eukaryotes. Things are happening much fast, so you need some kind of a robust mechanism. This polar force is driven by PAR-AB proteins, and the simple way to understand this is the PAR-AB protein is like a double well potential, and the ori can basically sit, ori can track this potential, fall in one of these throws, and then basically sit there, leading to their segregation. But what's breaking this symmetry of both ori not sitting in the same pole? That's one of the important questions that's not clear just by this framework. And so one of the questions is that. The second question is, we also see that the ori segregation mechanism is actually inhibited by structural maintenance of chromosome, SMC proteins. You have heard about cohesin. These are all parts of members of this family. So there's a bacterial family, and we see that happens even in SMC depletion. So basically what we did is got the structures, use this kind of a force field, and see this kind of a PAR-AB potential, and see whether segregation happens or not. And so we can get the structures. We can validate with imaging data. I'm not going over all this. Then basically what we need to do next is to actually recreate the replicating part, and this is something which contrasts the previous work. We are not doing it dynamically. We are doing it at every part of replication. So assuming that replication is a slow process compared to the polymer dynamics, I am just recreating the force field associating with every part of the potential using some symmetrization techniques. And basically now I can simulate what the replicated chromosome looks like. And so this is the original chromosome, this is the replicated part, and this region is what shows the inter-sister interactions, how the sisters are interacting. And you can clearly see that wild type has much less inter-sister interactions between the compared to the rest of the two. And so what you can also calculate is the inter-sister distance as a function of this replicated fraction. So this is, you can think of as time, early, mid, and late replication, and you can see wild type follows this, whereas mutants don't. So what's the underlying, you don't get to see cool videos. What's the underlying mechanism? Well, in the initial part, both of them sit at the same pole, but in the mid replication, basically they repel each other because of the structure, how all the proteins are acting, which is maintaining the structure. Now, thus maintaining that structure is what is driving this inter-sister repulsion, which is necessary to break the symmetry. And that's why they cannot sit at the same pole. They goes to this, but if either of these proteins you deplete, you lose this inter-sister repulsion, and that leads to missegregation. Missegregation of chromosome. Okay, so basically I showed you talk to you about energy landscape modeling and how we sort of use data to understand these aspects, and how chromosome dynamics is regulated by these proteins to sort of drive function. So that's it, and like to thank all the wonderful people I've had the opportunity to collaborate and work with, and of course the funding and you for paying attention. Thank you.
Thank you, Sumitabha. Questions? Vani?
So I was wondering, and you do these several iterations, so I was wondering whether you can have a sort of a landscape of the conformations which your polymer is attaining, and whether, I don't know whether it's relevant to your question specifically, because if we look at the single cell Hi-C, you have a lot of heterogeneity in the conformation. So whether we can correlate with that, given that specifically you are using the maximum entropy here.
Yeah, so our entropic constraints are only the means of the Hi-C. Those are the only constraints, because we only calculate the first moment of the distribution. And the second moment we can make a prediction and then check with the single cell Hi-C whether the kind of distributions is consistent or not. But the constraint is only with the mean, which is what you usually plot on the Hi-C map, is the mean. That's what is the constraint to get these ensemble of structures. And as you might also guess, like all the constraints are not always satisfied in every structure. There is frustration in the system. Some structures are only forming some, so there is sort of a antagonism in terms of what structures can form or not. So we see all those aspects in different subpopulations within the ensemble.
I had a question about the first part. So when you were doing this loop extrusion, you said you put it as an equilibrium force, but in the length compaction, but then you're also putting in an active force later on.
Yes.
So are you sort of double counting? That is part one. And part two is that activity you can put in many different ways. You know, there are these active, whatever, dipolar interactions and correlated noise and so on. Do all of them reproduce the same qualitative features in your results, like these extra bumps and so on that you get?
Yeah, very, very interesting question. So yes, in that case, one can set up the stage with certain activity and then train it from the data so that the double counting is already accounted for when you're doing maximum entropy. So in that way, you can use a baseline with the activity and then train on top of it to make sure that you are not double counting. In this particular work, we did not go the route of actually training from data. We were more interested in the phenomenological signatures. And yes, so the way we are looking at loop extrusion is not the dynamics aspect of it. It's more that it's compacts it longitudinally, makes it into a rod-like polymer. And this potential is basically mimicking that effect, that it makes it into a rod-like polymer. And then we can look at those aspects. So that's what we are doing in that part. And the different kinds of activity questions. So the way we are doing it is a monopolar sort of a force with memory, but they are all randomly oriented. So overall they cancel out. And if you do dipolar, there are many similar signatures. You see a lot of similarity in terms of these signatures. One of the things that is unique about this is if you use instantaneous dipoles, you will not get the features of like long time memory. If you have a memory versus if you have instantaneous dipoles give you, in terms of dynamics, it will give you an increased diffusion constant. It will not give you super diffusive signatures. Unless you have some kind of a memory, it will not give you super diffusive. It will mimic the effects of higher temperature. So that's something that we learned is, you know, the aspect of memory is actually important to drive these phenotypes of at what time scale, at what lag times do you see super diffusive light.
You can discuss during coffee. Yeah,
Thank you.
But over time. Thank you again.